Assalamualaikum. Good afternoon, doctors and everyone. My name is Nurliana Shafika from AS257, and today I will be presenting my FYP proposal titled Isolation of Antibiotic-Resistant E. coli in Hospital Wastewater. This work is supervised by Sir Helmi bin Yusuf. This is an overview of the presentation, so let's move to the first part. For background of study, antibiotic resistance is a growing global public health threat that makes treating infectious disease very challenging. Overuse and improper use of antibiotic accelerate the development of resistant bacteria. Among these, E. coli is particularly important because it is naturally reside in the digestive tract that can acquire virulence traits, sometimes causing severe disease like urinary tract infection, septicemia, and gastrointestinal illness. Hospital wastewater has been identified as an environmental source of antibiotic-resistant E. coli, containing pathogen, chemical residue, and antibiotic remnants, making it a potential hotspot for resistant bacteria. Secondly, problem statement. Despite the growing concern of antibiotic resistance, most studies focus on clinical isolate, with little attention given to environmental sources like hospital effluent. Resistant E. coli in hospital wastewater may enter the environment, posing a public health risk if not properly monitored. Therefore, my study aims to answer three main questions, which are: number one, which E. coli strain are present in hospital wastewater? Second, what antibiotic are these isolate resistant to? Third, are these difference in resistant pattern among isolated E. coli from different hospital wastewater sources? So to address this question, the objectives of the study are to isolate and identify E. coli from hospital wastewater, to determine their antibiotic susceptibility profile, and to compare the resistant pattern among isolate E. coli from different hospital wastewater sources. This study will provide new information on the frequency and pattern of antibiotic-resistant E. coli in hospital wastewater. The result can support better infection control and wastewater management strategies and guide antibiotic stewardship. Additionally, the study offers baseline for future environmental antimicrobial resistant research. For Chapter 2, which is the literature review, the key concept of this study includes antimicrobial agents, antimicrobial resistant, hospital wastewater as an environmental reservoir, and E. coli resistant patterns. Antimicrobial agents can be synthetic or natural, both playing roles in medicine but also contributing to resistance when misused. Antimicrobial resistance occur to antibiotics that were previously effective, often due to mutation or horizontal gene transfer. Hospital wastewater is a complex mixture of pathogenic bacteria, antibiotics, and chemical residue, creating a selective environment for resistant bacteria. Isolation of antibiotic-resistant bacteria from wastewater using culture-based technique allows us to study their characteristic and resistant pattern. Among these bacteria, E. coli is a common indicator organism. It is highly adaptable, can acquire resistant gene, and is abundant in hospital wastewater, reflecting resistant trend in a healthcare settings. Specifically, E. coli can develop resistance to B-lactams, tetracyclines, and fluoroquinolones, posing significant public health risk. Overall, this literature reviews shows a knowledge gap. Environmental sources like hospital wastewater have been less studied than clinical isolate, highlighting the importance of this project. For Chapter 3, which is the methodology, the study will be conducted in the laboratory using hospital wastewater collected from a designated effluent discharge point. The overall workflow starts with a sample collection and pre-enrichment to increase bacterial recovery, followed by isolation and identification of E. coli. The key material includes hospital wastewater sample, chemical reagent for gram staining, and biochemical tests for culture media such as nutrient agar and EMB agar, laboratory instrument like incubator, autoclave, microscope, and micropipette. Here are three variables in the study. First, independent variable, different hospital wastewater source. The dependent variable, which are presence of E. coli and antibiotic resistant profile. And the control variable will be the incubation temperature, incubation time, and media type. The experimental design involved selecting presumptive E.Coli colonies based on morphology, confirming identity via gram staining and IMVIC biochemical tests, and performing antibiotic susceptibility testing using the Kirby-Bohr disk diffusion method. All experiments will be performed in triplicates for reliability. Data will be analyzed by measuring zone of inhibition for each antibiotic and comparing resistance pattern across isolates and root source. This approach will provide insight into antibiotic resistance in distribution in hospital wastewater. We expect to identify specific E. coli strain and their resistant profile. This will help pinpoint environmental hotspot and pattern of antibiotic resistance in hospital wastewater. The potential limitation include sample degradation during transport, laboratory contamination risk, and variability in bacterial concentration. The mitigation strategies include aseptic sample handling, like using PPE, using icebox for transport, laminar flow cabinet, and the sterilization process. Lastly, performing replicates to ensure data reliability. The Gantt chart shows the estimated timeline of the project, including sample collection, laboratory work, data analysis, and report writing over the semester. Here are some references used in this study and research. That is all from me. Thank you, and all questions are welcome.